Good to see you, Marcella. Oh, you jumped
Wow, cool
that.
background.
Yeah, where are you? Or is that a virtual background? But that's ICM, isn't it?
That's ICM, exactly. Hello. Yeah, my
It looked,
computer is broken, so I cannot move from the open space.
mm. Oh, yeah, for a moment it looked like some sci-fi lab
Yeah, exactly.
with all the tubes and pipes.
Yeah, actually, yeah. And no, it's a real one. I'm in the real world. Yeah.
Hello.
Okay.
I do remember that part. Right. Okay. So it's established. It's 9:30. Eduardo has to officially ask questions now. Been waiting for this. Welcome to Neuroccino, everyone. 9:30 on a Monday morning. Today we have Lilith present a paper on, what was it? Structure and noise, connectivity and variability. If you
Mhm.
can share the link in the chat so we can all read along. Fantastic. And then take it away.
Okay, thank you. I'm sharing the screen. You see it well?
Yeah, if you can zoom in a little bit so we see it
Right.
full page.
Yeah, like this. Good.
Perfect.
Nice. Cool. Hello everyone again. Today I'd like to show you this paper published quite recently, January 21st this year. Uh, generally interested in variability, but in anatomical variability. So this paper, it talks about variability in dynamics, so in time, which is also something that I'm curious about. So I was pretty happy how the LinkedIn recommendation system work. So I bumped into this paper through that website, social media. So it was published by Terlo, Martini, and Helfrich in Neuron. So the authors are starting with the idea that humans can repeat the same type of behavior, complex behavior, repeatedly and precisely. But there is trial to trial variability. So for example, if you go and look at how pro tennis players play, they still can do the double fault when they are serving the ball, although they train the same action hundreds and millions times. So, but still they do this mistake. So there is some sort of trial to trial variability in behavior. And well, you can guess that that type of, variability comes from the brain variability, because brain is the organ that sends signal to your body and performs some complex action. Okay, so there is no question if brain has some neural variability. What is the question is that is this, as authors say, neural variability a bug or a feature? Is it something that hurts our performance or something that helps us to perform better? You know, and this is not really, seems like, a bit counterintuitive, because you might think like, well, obviously we need to decrease the variability so a tennis player keeps performing, serving the ball perfectly, every time. But if you go to the literature, one type of researchers, they say, research papers, they say that decreased variability is associated with better performance. But then you go to other type of papers, they say the opposite. Increased variability is better for performance. So what is the truth then? The authors, what they are trying to do, they're trying to reframe the results. So they are trying to um, explain with a bigger theory the evidence that we found in the literature. So the way they're explaining, they're saying, so first, it depends on the task. And second, there is regional differences, cortical regional differences in the magnitude of variability. Specifically, what they're saying is that sensory regions of like early like sensory regions like sensory motor areas, for example, they should have low magnitude of variability, while those regions more like associative regions, they should have higher variability. So there should be some gradient in this magnitude of variability, and it should depend on the task. So to kind of test their hypothesis, they tested 21 patients with epilepsy, and they recorded intracranial EEG. Super interesting for me. I don't know. So they recorded that in dynamic. And if you look, for example, into this, while they were recording those patients, they had to perform several tasks. The main idea behind those tasks is that they show some sample of like a queue, okay? And then after a delay period, they have to respond to the question. So very simple task. What authors did, they computed neural variability here. How did they do that? So they computed kind of standard deviation, but it's a bit more robust way to compute it. It's called the median absolute deviation, or MAD. So they computed MAD of the of the change from the baseline. So for example, if you look at here, you see 0. And if the values are negative, it means that the variability is decreased relative to the baseline. So, and if you have values above 0, it means that variability increases relative to the baseline. Okay? And here you see like a change of variability in time for the electrode in blue. And you see that when the Q is presented, okay, this the sample here, the variability goes negative, meaning that the variability shrinks. So they call in literature neural quenching this process when the variability decreases. But for example, if you go and look at the green ball, a green electrode over here, you see that the variability doesn't shrink. It just goes up, even during the presentation of the stimulus. Okay? So this is just the main framework, how they analyze their data. And the first question that they asked is variability, the magnitude of variability, task dependent. So, for example, if this electrode is always shows high variability in one task, is it possible that the same electrode will show high variability in other tasks as well? Okay, so is it task invariant or task dependent? So to do so, they calculated the average variability per electrode, and they computed pairwise correlations. Okay, so each dot is a electrode, average the variability per electrode. So you see here like positive correlations in the pairwise correlations, meaning that an electrode that shows high variability in one electrode will show high variability in other tasks as well. So it is like doesn't really depend on which task a person is performing. Well, at least in the tasks that they used in this study. Okay. So conclusion number 1, variability is task independent. Okay. Then they ask, okay, is there some structure of that variability? You know, like can we somehow understand why some regions show higher variability, or is it just random electrodes showing random levels of variability? To do so, they took Schaffer atlas, which has 400 parcels, 200 in each hemisphere. Then they ranked it from 1 to 200, where the 200 is the most associative area and 1 is the most sensory area. Okay? Then they split those 200 ranks into 10 equal units. So here you see like 10 equal units, and they just split this range into 10 to like average the variability across those parcels and ranks. Okay? And on y-axis, you see the relative variability. And what is happening, we have positive correlation between the rank of this axis, sensory to association axis is called SA, okay, and the variability. And what you are seeing here is that the most sensory, the more you go towards higher order in the hierarchy of the brain, the higher variability you get. So it means that there is a structure in variability. So this is the second conclusion. It's not random, like there is a specific structure to it. Next question that they asked, and by the way, it works, it is the same during the queue, okay? So when in this moment, so the variability here, there is hierarchy, and also there is hierarchy during delay period. So when there are no stimulus presented, okay? Okay, so yeah, the next thing that I ask, okay, we figure out that there is some structure in it, but why is there this type of structure? So they went deeper and tried to understand the mechanism why the association areas have higher variability. What they did is they, as I understand, they took other 74 patients, or maybe just expanded the sample, and they performed direct electrical stimulation while recording intracranial EEG. Super cool. So they did that, and when you are stimulating some region, you can get some, obviously you are getting a response, like in response to that stimulation. And that response, it's called corticocortical evoked potentials. And that response consists of two components, N1 and N2. So what is N1? If you look at this image, you see that this black knot is when you're stimulating like some region, and then you are getting outgoing like response signal. So this is a response that you are picking up in locally, and it reflects the feed-forward direct connections. Okay, while N2 happens later in time, and it is recurrent or like feedback connections, so they kind of come back to the node that you stimulated. So kind of you stimulate something, and then the signals goes away, and then it loops around and comes back, and that's what you are picking up. That's why it's later, because it has to travel through like some circuit. So they recorded and stimulated different parts, and you see that there are both components present no matter the region. So you get N1 and N2 everywhere. But what is different is the scale of N2. So the higher you go in the hierarchy of the brain, the amplitude becomes higher, bigger. Okay? And you can see over here, it's negative correlation. Well, it's negative because the amplitude is measured in a negative scale. So just it has to be negative if here, like the correlation. And again, that follows, yeah, this hierarchy. And you see the same pattern for the variability, but only for N2. So you see that N1 and in amplitude and variability, there is no correlation, so it's just flat. While for the N2 signal, so that represent recurrent feedback connections, you see the change. What does it mean? It means that in association areas, it seems you have more of those connections, possibly. And they tested it this way. They computed reciprocity index, which is a proportion of these recurrent connections divided by all the connections that are coming from that node, as I understand. So, and then you see that there is a correlation, which means that higher you go in the hierarchy, more recurrent connection you get. Okay? And they went further, say, okay, what if this relationship that we had over here, so the higher the hierarchy, higher variability, is mediated by the fact that there are just more recurrent connections. So idea behind it, like intuitive, is that, okay, so if there are more connections, recurrent connections, then the probability that it's will have more variability is higher because there are more like variables in game. So that's what they got. They also no, I mean they tried to understand if this relationship is mediated by this, and it seemed that their hypothesis was confirmed, that there is this relationship between um, uh, hierarchy and variability that is mediated by the amount of these recurrent connections. Well, and finally, they went and they tried to understand if this neural variability is somehow reflected in behavior. Okay, so the example that I gave in the beginning, like the tennis players, okay, it was kind of obvious that the brain variability reads to variability in action, but is it actually true, So that's what they tried to do, not about tennis players, unfortunately, but about some other tasks. So the first thing they wanted to see if there is some variability in the tasks. So they had like working memory, task which was like more task like more demanding. Okay, so it was a bit more difficult than the attention task, which was more like was perceptual task. So this but yeah, it was less demanding. And you see also it's reflected in behavioral, because in working memory task you have wider distribution, meaning that there was variability in behavioral, while for the attention task, it is more constrained distribution. It means that, they were answering like more consistently. Okay. And they try to understand if this behavioral variability is reflected in neural variability. And you see that only for higher order cortex, so associative areas, you can see on that only here, because you see that those two lines are kind of overlapping, and where here you have some differences which can be kind of reflection of those distributions that you see in at behavioral level. Okay. Next, what they did is, let me try to remember. 1 second. Yeah, then, wait. Yeah, then they compared, okay, the variability is reflected in, neural variability is reflected, behavioral variability is reflected in neural variability, but is it in the end like the key question, is it better, the variability, or is it hurting our performance? So the answer is more complicated because for the lower order structures of the brain, like sensory motor areas, you should have a decrease of variability. I don't know if you see there is like this gray rectangle. I hope you see it because on my screen it's not really visible, but there is this rectangle, which means like the Q, so when the sample was presented. So for the sensory areas, when the something is presented, if there is decreased variability, so it is lower, then you perform better. Okay? But for higher rank cortex, more you have the variability is better. Because you see like the correct answers are in pink and you see it's higher. But when you respond incorrectly for the task, the line is lower. And then maybe another thing, but they have just a lot of tests that I might miss something, but yeah, this dissociation. Well, yeah, so to sum up in general, is that first, what they found, variability is task independent. Okay, if an electrode shows high variability in one area, it will show high variability in another area. Then there is a gradient of variability that follows the sensory motor to association axis. Then the behavioral variability can be is reflected in brain variability, but only for the higher level functions order cortex in working memory task, for example. And for it's better you for a low order areas, you get neural quenching, so you have to have decreased variability to perform better. But for higher order regions, you have to have higher variability to perform better. Yeah, I sorry if the last part was a bit like messy because they did a lot of tests and yeah, maybe I missed one or two logical points, but I hope you got the main idea of this paper. If you have any questions, discuss. I don't know if it was too fast or not, but yeah.
Super exciting, and I'm very happy to see that Eduardo has the first question for you.
Wow.
Don't have a choice, don't I? Hi Lilith, thanks for presenting. So yeah, one question to start with is maybe I missed the beginning, but do they have a map or a graph where they show the distribution of the electrons they have?
Yeah.
I mean, because they talk about this like dissociation, this gradient between so say sensory primary and the more associative areas. But I wonder, is there at least slightly uniform coverage, or
yeah, this was something that so this one, as I understand, let's see, this is the intracranial
yeah, the code.
EEG coverage during electrical stimulation. So in this case, they have 74 participants, okay? And you see like the coverage is pretty, well, impressive. But what was strange to me maybe is this part.
Yeah, because there was also another plot, and I can't imagine so many electrodes goes into V1, for example.
Yeah, so in this case, so I had the same question over here. I don't know, because this part was based on 21 pharmacoresistant epilepsy patients. So this part, when I was talking about the gradient and so on and so forth, as I understand, they based on this type of coverage. And I also had the question, okay, but this
Yeah,
part,
okay. No, but it was
uh,
just I was just wondering whether they did some explicit testing or if they took into account.
no, but I think even though in this case it's not like fully covered, as I understand, that was replicated sort of in these 74 patients, as I understand.
Okay.
The question that I have actually about this is uh, if
Yeah.
Schleifer Atlas has 400 parcels and they ranked it from 1 to 200, it means that they just mixed the left and the right hemisphere together. So I was wondering if there might be actually the differences between the left and right hemisphere of the variability because it's not a mirror. Uh,
Yeah,
but
but I
yeah,
guess in the narrative of associative or sensory areas that can still go. I was just like a follow-up question, a short one. Do they what's their definition of like high order versus lower order cortex? Do they give one or what
so
is
this is the piece that talks about that.
yeah, but what do they mean by that?
So that axis, the gradient from sensory motor association axis was derived from systematic variation of structural, functional, and evolutional features as described previously. So I didn't go like into this
Okay.
paper, but they based that
Okay.
using like some previous research. So the division of the ranking, but that part was also a bit. Um, let's
Yeah,
see,
yeah,
what
no, I
is
was
this one
just wondering
called?
whether they just chose like say V1, S1, and so on as like a sensory motor.
Well, they are citing this paper, so I don't know. I don't know.
Yeah, no, okay.
Or maybe they meant this one. I don't know.
Yeah, no, okay, it's fine.
Yeah.
Yeah, I think Hervé
Mm-hmm.
has another question.
Oh, yeah, it's my time now. I was not late. Okay, thank you, Lilith, for this presentation. So very clear for, I think, a paper which is not so simple. So my question is about, so when you talk about gradients, variability, it rang a bell about aging. There was these theories about aging and theory. I don't know if you're aware. There was a guy, Cabeza, who was working on that, and he has one of his theory was the PASA model, where there is a posterior anterior shift where the variability it's not on EEG but it's on fMRI. So what it showed that with aging you have an increase of activation which follow anterior posterior gradient based on the primary and associative areas. And what he says is that it could be a compensation by the higher order regions trying to compensate for aging processes. And so in this case, higher variability in the associative area could be not so a good sign. So I don't know if they talk about it or if you see something in parallel to that in the paper.
I don't know. I don't remember if they start to talk about like in terms of aging, the variability in aging. But I think at the moment they're talking about in like adults. So I didn't also check the age range that they did, maybe in methods for these patients. Sorry for scrolling. Yeah, it's more like yeah, younger adults. In this case, even younger.
Yeah.
So yeah, they didn't. It was like, but I think it's really interesting to look also like at the aging population and see that the difference. Maybe it can be indicative or of some a faster degenerations or like a faster aging. It's very curious to look at. But no, I don't think I didn't read like everything, but yeah.
Because there is this debate about if this variability is more a compensation by the prefrontal regions, or if it is not, like you said, if it is a degeneration process where you have higher variability because the brain cannot support anymore the function. So there is this, what we see in
But also,
variability.
yeah. But also it may be, follow the same kind of gradient, and you just see like, okay, in a higher order, they get increase of even more variability, and that is already reaching the limit that leads to some pathology. So kind of maybe, yeah, there is some distribution of that variability that can be not beneficial anymore. But yeah, for that you need a different sample to check.
Yeah.
And then there will be differences in methodology, because I don't think you can get intracranial EEG with direct stimulation
Nah,
in
yeah,
that
it's not.
case.
Yeah, it could be complicated, but yeah, okay.
Yeah.
Thank you.
Thank you.
Thank you both. I have a question about the tasks. So you mentioned they also looked at cognitive functions, as in, I think, memory you mentioned, right? And attention, right. So I was wondering how generalizable this association is between the variability and the cognitive effects, and if that would also extend to other cognitive functions like language or social cognition or even naturalistic tasks in the end.
Yeah, so I think they chose this task for the simplicity, because it's happening during the, surgery, like the skull is open. So I don't know. But or maybe they use these tasks because previous literature based their conclusions about what is beneficial, high or low variability for performing a task. Maybe in that field they've been already using this task. So I don't know about
Hang on,
generalizability.
can I ask for clarification? So
Yeah.
the tasks were done during surgery, not just after implantation.
No, I mean surgery, I meant that the scalp was open, but I think it was done before the surgery. So
Okay.
no, I meant like surgery, the process of the opening the scalp and like doing the intracranial EEG and all of that. I think it's should be done before, right? Otherwise it's
Well, you usually test before to have a baseline, right? And then you test afterwards, and you can selectively also stimulate the different electrodes to see how that works.
I don't know. I should check better the procedure, but yeah, good point. But they didn't mention that. It was just my uh perception of how maybe it works, but I didn't read the method part exactly. The procedure was.
No worries. But you do have a point, like in clinical settings, time is
Yeah.
limited and a selective task is usually used.
So yeah,
Our
I don't
time is
know.
also limited.
Okay. But to finish,
Yeah.
I don't know if this is generalized. These results can be like replicated in like language tasks. But yeah, I think the idea was to take a contrast, one a higher order difficult task, like a working memory. But to me, it was like a bit more similar, the tasks. And also, we have to keep in mind that these were patients with epilepsy, so there might be some differences in, but yeah, this is, I think, a very nice paper. I really liked it. So
It's a super exciting paper, but you're right. So given that they're patients, you also have usually you have a higher implantation rate in the temporal lobe, and therefore memory is, for example, a better cognitive task of interest. Right. Time's up. Good luck fixing your car by printing spare parts. I have done that in the past. You're right. Thanks for presenting this exciting paper. Have a really cool week and see you again next Monday for another Neuroccino. Bye.